Okay, everybody wake up. Bear with me here. You're going to see this again on the file. Pay attention. I'm not just doing this to do it. Okay? So, get into the problem. The problem is, you've got a cross between gray body, normal wings, with blackness visual reason. Gray is dominant as the black. Normal long wings is dominant as the visual. So, what would you expect? How many of these flies would you expect to be the gray body? That's all I'm asking. I'm not asking about the wings right now. How many would you expect?
Be gray or black?
You expect half and half, right? So you expect half to be gray, half to be black. I'm asking how many were actually gray?
955.
Good, that's it. So we know 965 are gray. That's the wild type. And then the other grays were 206. So 965 plus 206. Okay. How many were black? Good, 944 plus 185. Add those up, and those two numbers are going to be pretty close. So therefore, the answer to the question is, yeah, this appears to support. There's no reason at this point to reject the hypothesis that segregations work. The same thing for vestigial wing versus long. How many were long wing? We're expecting the same thing, because it's essentially the same cross. Half should be long wing, half should be vestigial. So how many should be long? Or how many were long? 965 plus 185. Excellent. And then how many were vestigial? Right, so good. So that's 944 plus 2 and 6, and again about half. Okay, so C is correct, A is correct, B is also correct. The correct answer on that one is, we got them all over the correct, so that's going to be confusing all over the one. That was expected to be a humdinger, so basically I made that a bonus. If you got it wrong, it didn't hurt you. If you got it right, you got extra credit. That's three extra credit. Okay? So, that's where we say everything else went pretty well. I think the questions that came late at the end, the next problems were done for the most part pretty well. This one was kind of tough because you hadn't really seen much of it, so you had to kind of work that out on your own. And a lot of people hadn't really well. It's pretty impressive. So all in all, I'm pretty happy with what's going on here. But I will say one thing. There are a lot of people who really chunk this exam. This is surprising because typically then, when we look at this group, usually when I hand test back, I'm missing 6 or 7 out of this mistake. We don't feel this way. Those people are not here. However, many of you work from this class this group is moving. I notice a very, very strong correlation between the quality of your answers and whether or not you're on class. So, yes, it's a good class. Yes, I know you. I know who you are, and I know when you're not here. So I can do that correlation. If you're not done the class, you know who you are. That's why you failed this exam. And that's the only reason. No one in this room could fail any of these exams. No one. There's no excuse for it. None. No one in here has a brain that's too small to handle it. No one in here is so unmotivated that they can't handle this material. No one in this room should be getting a D. Everyone should be getting C or above. And I know that because I know you. Even if you're not in my lab, I know you. I've talked to your lab instructor. So, this is a great group. It's one of the best groups I've ever had. There's no reason that anybody should get a D in that group class. None. If you're getting an A or B, fantastic. If you got the 108, fabulous. Great job. If you got above a 95, fabulous. You're doing exactly right. Keep going. If you're low, first, make sure you're in class. Second, make sure you're on top of everything. Third, if you're still doing all that and you're still struggling, come and talk to me. There's techniques to get you over the problem. Many of you are having trouble, and it's because you're lacking confidence, or it's because you're thinking that somehow there is one way to study when that way isn't particularly effective. Right? So there's a lot of people who did not so well on this exam, and they were here. And that was largely not because, again, the brain is a problem. The problem is that in some cases I'm anticipating it's because the studying is not working right. In other cases, I know pretty much for a fact that it's lack of confidence, that it's totally unwarranted. Everyone in here should not only be getting a C or above, everyone in here should be confident that they're going to get a C or above. Is that good? right, good. That's the end of the sermon for today. Let's go ahead and move on. All right, so we're going to start now with the next unit, the third unit. The third unit lectures are currently not on the new site, they're on the old site. Download there. Now what we're going to do is we're going to address a whole different topic. We're addressing what we've been talking about before are issues associated with how it is that the trade has paired offspring and the consequences of that for the population, consequences of that for the family. Now we're going to look at a much more detailed level. What we're going to do is we're going to look specifically at how is it that a particular gene causes a particular trait. For example, people who are born with cystic fibrosis. I know a couple of different things about that. First of all, I know that both of their parents must have been heterozygous. You know that too. Cystic fibrosis is a recessive trait. In some ways, like some of the other diseases we looked at, for example, Duchenne muscular dystrophy. But unlike DMD, it's not sex-linked. CF is not sex-linked. It's sort of like thalassemia, although unlike thalassemia, there's no minor form. It's just you either have it or you don't. So I know then that if an individual has cystic fibrosis, that means that their parents were heterozygous. They can't have had cystic fibrosis because I know another thing is, and that is that if a person is born with cystic fibrosis, they're going to die before they reach the age of 12. Almost all the time. They can survive a little bit longer, but even then, chances are their bodies are not going to be in a position where they can support any kind of pregnancy if they're female or reproduce if they're male. So people with cystic fibrosis don't reproduce. My question is, what is happening in that person's body that is causing their deaths at a very, very young age? And entirely because they're born with it. It has very little to do with the environment. In this case, you're born with it or you're not. And that question then is, what is it that they're born with that's causing the problem? Here's a couple other things I know. I know that the problem with cystic fibrosis is a particular protein that is made incorrectly by individuals who have it. So people who have cystic fibrosis can't make a particular protein in their lungs called a chloride channel. And that chloride channel is non-functional, and that's what leads to their death. So there's two issues here. What is it that causes the problem with the chloride channel? And what is it that causes the problem with the chloride channel causes to destroy their ability to ventilate their lungs? So in other words, how does that genetic change affect the phenotype? All of that follows from this paper. It was published in 1953 in the journal Nature. Molecular structure of nucleic acids. This was the collection of papers, and the title of the paper is A Structure for Deoxyribonucleic Acid. And this was written by these two guys, right here, James Watson and Francis Crick. That's Francis Crick, that's James Watson. Both of them are still alive, although they're much older. Okay, this was done again in 1953. And this paper is one of the most important papers ever published, probably the most important paper ever published. It's a simple paper, it's only a couple pages long. And all it did was give the structure of this molecule right here, which they're pointing at, or looking at, or whatever, in this very famous paper of their model of DNA. This was done in 1953. They built this model in 1953. In 1953, the best microscope we had couldn't even see mitochondria. So mitochondria are thousands of times bigger than DNA. So no one had ever even come close to seeing DNA. The electron microscope had just been invented when this paper came out, and they had no access to one. So they did it entirely chemically. They used nothing but chemistry, and they figured out that model. Now, since then, our microscopes have gotten better. Since then, our chemical techniques have gotten better. Since then, all of our scientific techniques and technology have exploded in this field. And all of that has shown us, one of the more astonishing things, is that this model that they're pointing to right here is right in every single detail. We have never once, in the intervening years since 1953, with all of our new technologies, ever had to revise their model. They nailed it. They absolutely nailed it. They got it right in every single atom. Every single bond angle, everything is perfect. It is absolutely done. They did it because they were very, very careful and they had some help. These guys got the Nobel Prize for this with one other guy, a guy named Wilkins. And Wilkins had been doing some work with them to try to figure out what the crystal structure was of this molecule here, which is the molecule that they worked out. Now, the story is really kind of interesting. What happened was they were pretty close to getting the structure, and it had to do with getting the bond angles in the molecule all correct. And a guy named Linus Pauling, who was famous already by that time, he already had the Nobel Prize, had come up and said, "I've got the structure of the triple helix." And when he came out with that paper, these guys were like, "Oh, crap, that's it. Pauling's got it right. He gets everything right. So he's got it right." And they looked carefully at the paper and they realized, no, he's wrong. We know from some of the, at least, he might be a triple helix, but his data don't show that. So they started working very, very feverishly trying to find the structure. And while they were working with Wilkins was working with this woman named Rosalind Franklin. And at the time, she was the top person in the world at a technique called X-ray crystallography. And this is about the best you could do to see a molecule. What you do, you take the molecule and you crystallize it. You take it out of solution, crystallize it, and then shine X-rays through it. And the X-rays then will be giving you a pattern like this one here. This is the most famous of them. I'll explain why in a second. And the X-rays then give you a particular pattern that you then capture on a photographic film. And it's equivalent to essentially doing the following thing. You have a very complex three-dimensional structure. You're trying to figure out what its structure is. And you can never see it, ever. You can't ever look at it in any angle. And all you have is the following. You have data from the following experiment. The structure is really big. Imagine it basically, to fill this room. You hang the structure up really high and take a gigantic vat of marble. Put that gigantic vat of marble above the structure and drop all the marble all at once on the structure. And bounce off the structure and bounce here and then so on. And you mark the location of every single marble when it hits the ground. And then you take those data, and from that, you figure out the structure of the data. That's what this is. That's essentially what this X-ray crystallography is. It's shining a bunch of photons through this molecule, and they scatter. They bounce off all over the place, and then they hit this photographic plate. And that's the pattern that you get. Now, Franklin had been working on this. He's been working on proteins. He wasn't really trying to get the structure of DNA specific. He was trying to take good X-ray pictures of it. And so Wilkins, one day, the guy who worked with these two guys, one day went to her lab to see, because she knew she was trying to get this picture that's right next to her face there. By the way, it's really irritating. I don't know why it is. Men scientists always look like dorks. Women scientists don't. It's just not true. But whatever. She was out of her lab. Wilkins walks in. And she's gone to a conference. He walks into her lab and he says, "Oh, okay, I know she's been working on doing X-ray crystallography of DNA." And one of the grads says, "Yeah, I know. Here's the picture." And he shows Wilkins that picture. And Wilkins looks at that and says, "Is that Robert?" And the grad says, "Sure." So Wilkins goes back to these guys and says, "Hey, look, this is Rosie's," that's what they call the Rosalind Franklin. "This is Rosie's latest picture from this X-ray crystallograph of DNA. Watson and Crick both look at that, look at each other, and realize they just got the Nobel Prize. They were just handed the Nobel Prize. They looked at that and both understood immediately that it's not triple helix, it's double helix. That is the picture you get from a double helix, not triple helix. Why? You have to get into huge amounts of physics to see it. But they saw it, they knew it. So then they write this paper. Wilkins writes another paper that comes after it. They submit that. And many, many years later, they get the Nobel Prize. Okay? Rosalind Franklin did not get anything. She got no Nobel Prize. That's not true. It's not that she didn't get it. She got a lot of recognition, but she never got a Nobel Prize. And so you're going to hear a lot of issues with this. This comes up a lot of times in scientific philosophy classes, scientific society classes, and I just want to leave you with a couple of things. That oftentimes the whole story is not given in those classes. I just want to point something out. The Nobel Prize Committee said they didn't give her the Nobel Prize because she had died in the years. Tragically, she got, between the time that they published this paper and the time that they got the Nobel Prize, she contracted ovarian cancer and died from it. So the Nobel Prize Committee just does not give posthumous Nobel Prizes. That's one of their rules. In fact, you guys might have known about this. This latest Nobel round of Nobel Prizes had this. The guy was awarded the Nobel Prize on Monday. The university was told that he got the Nobel Prize on Monday and he died on Saturday. So now they're trying to figure out should they give the Nobel Prize or not. They are going to give it to the family. So yeah, I didn't know that. So that's the issue. She had died long before, so it wasn't that close an issue. Okay, so the Nobel Prize Committee basically wants you to say, yeah, she survived, we'll be giving her the Nobel Prize. But all the citations and all the work that was done before that, she was never mentioned. So, these guys came up with the Nobel Prize before she died, and she wasn't mentioned in that. So my take on it is that she would not have gotten the Nobel Prize. Right. Now, another thing that often isn't heard is her voice. She was asked specifically, when she was alive and when these guys were first nominated for the Nobel Prize, do you feel you should have been nominated? Guess what her answer was? No. She said no. So, again, you know why? She might have been motivated because she made a public statement and so on. She may not be expressing herself truthfully. But nevertheless, she said no, she didn't believe that she could have gotten it, or that she should have gotten it. And she was asked specifically, did she believe these guys should get the Nobel Prize? And she said yes. There's no doubt in my mind that they should get the Nobel Prize. Now, it's also been sort of described as a big war between her and these two. There's a big fight. And it turns out, in fact, that is absolutely false. This guy right here is a first-class jerk. He is just one of the most horrible human beings I've ever met. He is completely nerdy, but he's a wonderful guy. Great guy. And there wasn't really any kind of animosity between these groups, but I just want to point something out. While she was dying, a very new technique that we now use standard on treatment of ovarian cancer is radiation treatment. And she was, at the time, there was no radiation treatment that was available except in Berkeley, California. And this guy packed her off on an airplane and took her to Berkeley to get treatment at the one radiation treatment facility in the world, and he paid for it. So there's clearly a lot of strong friendship. So anyway, I just want to point that out because when you take a philosophy class or you take another humanities class, you're going to talk a lot about this. And they put it in the context of science and society, and it's just important to get her voice. Listen to what she says, read what she says about it, as well as what everybody else says. Okay, so at any rate, why is it so important? Well, here's why. Because of that discovery, we now can do this. This is called fluorescence in situ hybridization fish. We want to figure out if a kid is sick, or we want to figure out if a kid has Down syndrome, we want to figure out if a fetus is male or female. It's really easy now. We don't have to do very much. All you got to do is this. You take a cell out of the amniotic fluid, and you probe it with a particular probe that you can construct because you know the structure of DNA. And the only way we know the structure of DNA is because of those guys working on it. And that probe will fluoresce if you shine it under light. So, if, for example, you're trying to determine is the kid, is that fetus male or female, you've got one cell for the fetus, and you probe for a probe that will bind to the Y chromosome, and only the Y chromosome in this particular spot. And it'll fluoresce, say, red. And then you put another probe in there that will fluoresce blue, but it'll only bind to the X chromosome. Then you just put that in, you don't have to kill the cell, you don't do anything else. Put it in a microscope called a confocal microscope that shines the light to the fluoresce. If you see two red dots, you're looking at a female. If you see a blue and a red dot, you're looking at a male. If you see two red dots in the blue, you're looking at bicolor. It's that simple. Really fast and really easy to do because of what those guys were able to do in 1960. All of that technology derived from that. Here, you're looking at the brain of a rat that has been damaged artificially to simulate a stroke. Strokes, damage to the spinal cord, anything that occurs in the neurons, anything that occurs in nervous tissues, you probably know, is irreparable. So if you've got somebody who's got a severed spinal cord, they're not ever going to use that essentially anything again. You might say, well, wait a minute, that's not true. I know somebody had a spinal cord damage and they got their leg motion sent back, or they were moving it. That's not because the nerves knitted. The nerves don't knit. It happened because the swelling went down, and it allowed the nerves to start functioning. But if nerves are severed, nothing you can do. The strokes are devastating. It's a tissue that's gone. You're not going to get that tissue back. The brain has to adapt in a different way. In this case, though, in that simulated stroke, what they're studying are the the green cells. Green cells are fluorescing what's called green fluorescent proteins, and they are marked. They are stem cells. Those cells are stem cells, and so you'll learn later exactly what stem cell is. They're seeded into that stroke area. And here is exactly the same brain some weeks later. And you see the cells that are fluorescing are the same cells that they originally had as stem cells. They're no longer stem cells. If you look at this picture, what you're looking at are a bunch of neurons. Those are perfectly differentiated neurons. And this brain tissue, the architecture is perfect. The brain regenerated. All of that technology is available simply because of that 1953 paper. All that derived from that paper 1953. All that paper did was give the structure of DNA. All of this things from that. This sheep is a Dorset sheep. She was born out of the womb of this Scottish black-faced sheep. So this is her mother, kind of. This sheep shares no genes whatsoever. Not one gene is shared between this sheep and its mother. All this sheep's genes come from a different Dorset. And those genes came from a mature cell in that genetic mother's udder. It's equivalent to doing the following. Take you, and I'm going to take a blood sample. And from that blood sample, I'm going to take a couple of white blood cells. I can only need one. We need one white blood cell. It could be any cell in your body. I don't care what it is. Give me skin cell, whatever. I'll take one little cell. I'm going to take its nucleus. I'm going to put that into another cell that will act like an egg. And then what I'm going to do is I'm going to convince that hybrid cell that it's actually a zygote. That's the tricky part. Once you get it back on the zygote, it then will form into an embryo, will form into a fetus. We talked about that before. And then you end up with a clone of you that is whatever age you are now, that many years younger. Your genetic twin, absolutely exactly the same as you, except 20 whatever years younger. Okay? We can do that because of that. We can do a lot more than that because of that. So that's why I say this is the most important paper for public. All of our biotechnology, $1 billion of dollars industry comes out of that 1953 paper. Watson Crick got the Nobel Prize, and he didn't have money. So what is it that they discovered? Well, what they discovered is given to you here. We're going to come back to this in a second. What they discovered was the structure of DNA. And I want you to learn it. Because if you want to understand all those technologies, if you want to participate in those technologies, if you want to be on the leading edge of any of those technologies, and I know many of you would do, you've got to know the structure of this molecule, atom by atom. If you're not going to be on the leading edge of those technologies, there is nothing in biology that you can do that does not require you to know the structure of this molecule. This molecule is the center of all biology. So, I want you to learn it atom by atom. It starts with a molecule of sugar. And that molecule of sugar is called ribose. So what I want you to do is to first learn the structure of ribose. Okay? All our DNA can be based on this sugar. I want you to know the structure of this atom by atom. Okay? So I want you to be able to draw it from memory. To start, start with an oxygen. Right here. Put an oxygen there, and then what you're going to do is you're going to connect this to 5 other carbons. 4 of the carbons are going to be in a ring like this. Okay? So that forms this ring structure, and then the 5th carbon is hanging out over here. And everything else is hydrogen and oxygen. Now watch how I draw this. It's important. We did this last week with the glucose. Now we're ribose. Sticking off of here is an H. Sticking off here is an OH. Sticking off here is OH. And an H here. H here, an O, an H here, an H here, O, H, H, H. That's ribose. Memory. Now, when I draw it, I'm going to draw it in a sort of a simpler way. So I can draw it this way as well. I can draw it like this. Put the oxygen here, and then draw a pentagram. Okay? And then, notice I'm not putting C here, or here. This is the organic chemist way of drawing things. And just basically, since everything in organic chemistry is made out of carbon, you don't draw the carbon. So every place where you see a corner in this pentagon is carbon somewhere about the oxygen. And then I've got now oxygen, hydrogen, hydrogen, oxygen, hydrogen, hydrogen, oxygen, hydrogen, hydrogen, oxygen, hydrogen, hydrogen, and so hydrogen, carbon. And then here is going to be H2OH. That's the way you simplify that. Okay? So these two things are equivalent. That's the so either way is correct. Either way is correct. OH is the underlying key. No, notice I don't have them here. You don't have them here. Yes, this shows this is the complete structural formula. This is the simplified structural formula. Now notice, see this H2COA. Nobody sees H2COA. Okay? No one in the world sees that. What you see in your head is more like this. CHHOH. That's what you see in your head, sort of. Actually, when you write this, nobody sees these. What you see in your head is the actual three-dimensional object. What you've got here is a three-dimensional molecule. So you got to get used to taking that three-dimensional molecule and drawing it, and still, even though you're drawing something in 2 dimensions, still seeing it in 3. And there are techniques of doing that. There are techniques to help you do that. For example, in organic chemistry, you'll learn the so-called chair structure of glucose and so on. There are techniques of doing that here too, but if you draw that, just be thinking, remember, you built those molecules last week. So same sort of deal. Don't need to draw. Right here, you're exactly right. There's nothing else to do. That's the item. We call it. Okay? Alright, so now I want you to compare that to this. Oh, bang, 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 bang. Look at that molecule compared to the other one and tell me how they differ. Yes, so you're missing something here. See it? No H here. This molecule is a molecule called ribose. That's I have up there. This is a molecule called missing ribose. Okay, oxygen. Right. The only difference between ribose and deoxyribose is that missing oxygen. Now, which carbon is it? Well, so it's the one in the lower left-hand corner, to the lower right-hand corner, it's three-dimensional object you flop around right. So that's hard to describe. So what we're going to do is this. We're going to come up with a number of these. When we talk about the carbons, we're going to have to do a lot. We can see exactly which carbons we're talking about, and we can state it directly without any ambiguity. Here's how you do it. Start with the oxygen in the ring. And you notice, it's easier probably to see it here. Look, this oxygen is connected to two carbons. And those are the only things it's connected to. There's a carbon here and carbon here. These carbons are asymmetrical. Why are they asymmetrical? What makes these two things almost symmetrical, but they're not quite? C1. Now, this is attached to an OH. This is attached to a C. See that? So this carbon here is outside the ring, and it's attached to a carbon that's inside the ring. It's otherwise very closely symmetrical with this carbon. All right, start there. Orient yourself on this oxygen in the ring. Look for this carbon that's attached to the carbon outside the ring, and then go to the other one. That's carbon number 1. On every single sugar. Any sugar you ever see, that's what's the 1. Okay, so this carbon here, right there, is carbon number 1. Now when we draw it, that's the standard way of drawing it, is to draw it this way. But you got to realize, this is a three-dimensional object. So I can flip this over. You still have to be able to find carbon number 1. There's carbon number 1. Then you number them in order as you go around. So if that's carbon number 1, this is carbon number 2, carbon number 3, carbon number 4, carbon number 5. Okay. It's not always clockwise. You flip, right? You get the three-dimensional object. Remember, so think in your head. Here's what when I draw this, what I see in my head is very similar to those models we put together last week and last. That's what I'm seeing in my head. And this, I don't see this as having a right or a left. This molecule doesn't have a right or a left. This molecule just seems to be flipped to the upside down in any direction. So to find that one, I've got to find the oxygen in the ring, go to the carbon that is outside the ring, and then go opposite. And that's carbon number 1. The other way to do it, find the carbon outside the ring, call that carbon number 5, move number backwards. That's the way it works. This is unambiguous. Everybody in the world, you can be talking to somebody from Tuba, and you don't understand a word they're saying. You go on that, you'll know the carbon is exactly the same way. Okay? Every geneticist in the world uses this notation. So which carbon is missing the oxygen on deoxyribose?
2.
Carbon number 2. See, that's why we do this. Because there's going to be things like which carbon has this, which carbon doesn't have this. Okay, so yes, carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. Okay, so that's number 2. Alright, so now to make DNA or RNA, either one, I start with one of these sugars. If I'm going to make DNA, I start with deoxyribose. If I want to make RNA, I start with ribose. But otherwise, they're identical. So what I'm going to do is I'm going to build nucleic acid. Build nucleic acid, I'm going to make what we refer to as a polymer. And a polymer is like this. A polymer is where you take a bunch of objects which are similar. They don't have to be identical, but they're similar, like these pens. These pens are similar. They're not identical. This one's red, this one's green, this one's black, but they're all basically the same shape. And what I do is I take one of these objects and I connect it to another like this. And now I have what's called a diamond. And then I connect it to another one, and make it a primer, then a tetramer, then a trimer, and so on. And I hook all these things together. That's what a polymer is. That's all a polymer is. It's basically a repeated unit over and over and over again. They're covalently bonded together. Each of the single units is a monomer. So polymer is a chain of monomers. In this case, the monomer is of the 10, and the polymer is this chain of 10. And so polymer is equal to a chain of monomers. DNA and RNA are both polymers. In this case, the monomer has a name. The monomer is called a nucleotide. So nucleic acids, which include RNA and DNA. Nucleic acids are polymers of what are called nucleotide monomers. Right now, what I'm going to do is build a nucleotide for you right now. And then we'll build the DNA or the RNA from that. Okay? So, to start, I take a ribose sugar or a deoxyribose. It depends if the nucleic acid I'm building is RNA, I take ribose. If the nucleic acid I'm building is DNA, I take deoxyribose. Right now, that doesn't matter. All nucleotides have the same structure. They have the sugar at their core. To that sugar, I attach two different groups. One group is referred to as the phosphate group. So I attach a phosphate, and a phosphate looks like this. A phosphate is a phosphorus atom with covalent bonded with a double bond to an oxygen, to another oxygen, to another oxygen, to another oxygen. That's CO4. Now notice, I got a negative charge here, negative charge here, negative charge here. Those things that chemistry should recognize this. What is this? Phosphate, yes. If I put hydrogens where those negative charges are, what would I have? Phosphoric acid. Exactly. So this is the salt of phosphoric acid. That's what this is. Okay? So if you're not sure what the salt of phosphoric acid is, you want to do one to learn. Okay? If you haven't had that, don't worry. But this is the phosphate, CO4. It's not phosphorus. It's got the oxygen. I attach that. Now looking at this, can you tell me which carbon have I attached to phosphate? Good. Carbon number 5. I always put the phosphate on carbon number 5. Okay, so when I'm building nucleotides, the phosphate is always on carbon number 5. Okay, so now I've got 2 of the 3 elements. Third element, not really element, third moiety, third group, is going to be put on carbon number 1. And it is a base, and we can talk about acids and bases here in just a second, I'll make sure we know what that is. But it's also an organic base. Organic means what again?
It contains carbon.
It contains carbon. But it also contains nitrogen. So it always has nitrogen on it. In fact, Whenever you see an organic compound with nitrogen on it, you know it's a base. Nitrogen on an organic compound is almost always a base. This one's nitrogenous. So it says it's an organic base, but it's got nitrogen on it. Okay, so tell me what makes something an acid in aqueous solution?
Hydrogen.
Hydrogen, right. Anything that increases the hydrogen ion concentration in aqueous solution is an acid. You need to know that. Okay, so acid. An acid is something that increases the concentration of hydrogen ion. Now, I'm using the shorthand notation from chemistry. This notation means concentration of. If you weren't aware of that, put that in your notes. This says concentration of whatever's inside the bracket. In this case, concentration of hydrogen. Notice not concentration of hydrogen, but concentration of hydrogen ion. So anything that increases the concentration of hydrogen ions is an acid. You can apply phosphoric acid to that. You can dump it in there all the time. Okay, so what's the base?
OH.
It increases what? Good. OH. It increases this ion, hydroxide, OH. Another way of looking at it is 4 decreases hydrogen ion concentration. You know what it works. Which means this also is this, 4 decreases hydroxide. Take the solution, those two things are always going opposite direction. Okay? So this is a base. You put this in aqueous solution, it absorbs hydrogen. Okay? And now you've got those two things connected. Sugar, which could be ribosaccharides, base, and the phosphate. That's your nucleotide. That's the monomer on which I'm going to build DNA and RNA. Okay, now the nitrogenous bases that you can add, the available bases, are pretty limited. And it depends on whether you're now it starts to depend on whether you're making RNA or DNA. If you're making DNA, many of you know this. So to make DNA, you have available what bases? There's one. A stands for adenine. Right. So one of them is adenine. Anybody know any others? So good. So cytidine or cytidine.
One.
No, you also have thymine. And I see that. And the other one was one. Now, these are just the bases for DNA. I take the base, and that's the A. That's the thing that's labeled A there, the adenine. I attach adenine to the sugar and the phosphate, and I have what's called adenosine. Adenine is the base. The base plus the sugar and the phosphate is adenosine. Okay? So, same thing with these others. Sure. Adenine is the base. When I attach the base to the nucleotide, the rest of the nucleotide is called adenosine. Okay, so this is the base. The nucleotide that has that base on it is adenosine. The nucleotide that has guanine on it is guanosine. The nucleotide that has cytidine on it is cytosine. Okay? So technically this is thymosin. Alright, so there's your nucleotide. RNA is different. Okay, so to make RNA, what's the difference? Anybody know? Good, perfect. So this one has adenine. Come on, say A. It has the guanine. It has the cytidine. But instead of thymine, it has a molecule of uracil. Uracil is very similar to thymine. Not exactly the same. So it's slightly different. And why is it that RNA takes uracil to thymine? It has to do with physics of losing that oxygen. Losing that oxygen on carbon number 2 has an effect on the bonding energy to carbon number 1. And the bonding energy to carbon number 1 is more thermodynamically favorable in RNA to have uracil if you've got that oxygen on carbon number 2. If you can't see it, don't worry. Nobody can really see it. You have to do math. The math tells you that's what's happening. Just look at the molecule, you can't see it. Okay? But that's the reason. Okay, So that's the difference between DNA and RNA. Otherwise, very similar. Okay? There's another difference which I'll get into in a second. Now, what I want to do is start to make polymerase. What I'm going to do is make DNA. I'm not going to make RNA. RNA is simpler. So I'm going to make DNA. To do that, I got to take a bunch of these and chain them together. So what I'm going to do is take another nucleotide and put it together with this one, like that. So I get what's called a dinucleotide. And the dinucleotide now has, notice the first one is the one we had, and the second one has cytosine on it. Or the second one is cytosine, so it's cytosine on it. And you got the deoxyribose, the phosphate, and the cytosine. So all of that then is your next nucleotide. Now look at that carefully and tell me, I have made now a bridge. And you notice that the bridge goes from one carbon to another through the phosphate group. To what other carbon is the phosphate on the cytosine attached? So the cytosine and the adenosine are attached through that phosphate. What is the carbon that that phosphate is attached to on the adenosine? Carbon number 3. Exactly. See that? So this bond goes from the carbon here. What carbon is that?
5.
5. That's carbon number 5. It attaches to carbon number 5 to carbon number that one. Carbon number, I have a better shot. That's carbon number 3. Okay? So I'll draw it. It'll make it easier. So now, here's my deoxyribose. I want to attach that to another nucleotide. Now, speaking off here, replace the H with what? On carbon number 1, it's no longer an H, it's here. What's there? Carbon number 1? Base. You got it. There's the base and base. There's the base. And now the phosphate is attached here. Right? So I have the carbon, attached to hydrogen, and then through the OH group, I'm attached to this phosphate. Like this. Okay? Now, here, what I do is I attach phosphate to this. C-O-O, like this. And then, what do I have to put on there? Make the next nucleotide down. What goes here? The carbon, right. So this is attached to carbon. Which carbon is that?
5.
5. You got it. Good. There's hydrogen, and then this is attached to the rest of the nucleotide. Okay? What attaches here? Base? Good. Which carbon is this? 2. So what's attached to it? What you going on what? H here and H here because it's deoxyribose, not ribose. Okay, which carbon is this? 3. What goes here? H and OH. Good. Okay. Don't check out now, folks. Stay with me. Okay. There's nothing really weird going on here. I'm just drawing pictures. Just follow the picture. And you did these molecules last week. You haven't intuitive sense of how these things are working. This goes with an H here. Okay? Now notice, here's the bond. That's the bond I'm talking about. So here's nucleotide 1, here's nucleotide 2. On this picture, this one is this. A or 2. Okay, and then this one is G. C. Okay, so now, the bond I'm interested in is right here, this. I'm connecting carbon number what?
5.
5. To carbon number what? 3. So this is a bridge between the nucleotides. It passes through the phosphate group. And this bridge is called a 5-to-3 phosphate bridge. And I say 5-to-3 what? What's 5-to-3 referring to? The carbons to which those things attach. You got it. Yes, you can bridge these in different ways. You can use the phosphate to bridge in different ways. But in DNA, you use this. Okay, now, here is called a phosphodiester bond. That's the correct term for it. This is, in fact, an ester bond this way and an ester bond this way. It makes you a diester. What's an ester bond? What's a phosphoester bond? In organic chemistry, it's just more trouble than it's worth to try to get into right now. If you're going to take organic chemistry, you know people what that is. Right now, you can just call it a 3,5-phosphate bridge. What I'm more interested in is making sure you understand that's where it happens. 3 to 5. Okay, good. So you've got that. And now I've got a dinucleotide. Okay, so I'm constructing the dinucleotide. Now, to make this into a polymer, I just have to put much more on it. Okay, so that's fairly straightforward. All I got to do is just take this, attach another one, attach another one, attach another one, attach another one. Yeah. Yes, it should be 5 to 3 or 3 to 5. In this case, I say 5 to 3 for a reason which I'll describe in just a minute. The short story is a cell reads this from 5 to 3 to 5. You could say 3 to 5, but we couldn't say 5 to 3 because we're talking about how the cell takes these balls. Yes. Okay, so good. Let's count carbon. My finger right here is on what atom?
2, 1, 3.
That's an oxygen. There's the oxygen. Okay? Carbon number 2, 3, 4, 5. Okay? Okay? And so, yeah, it's, when we draw the shorthand, it's hard to see where it's from. Okay. Any other questions? Have you break down? Yes. Good. The question is when the carbon is binding to the phosphate, what part of the carbon here, what was attached to the carbon is now part of the phosphate, that oxygen. That was the OH group. The H comes off, and the O is part of that phosphate. Okay? The hydrogen is released through the reaction, and this is what's called dehydration synthesis. Remember last week when you did your lab report, and when you did this, you had to take OH groups off one molecule and H off another to make water. Well, this H went to make the water. Okay. Now, the other thing is this. If I take this out of solution and crystallize it like Dr. Franklin did when she was making those X-ray crystal graphs, what happens is this becomes like phosphoric acid again. So it picks up covalently a hydrogen. Okay? So this is what happens when it crystallizes. So I take it out, I get rid of all the water, I dry it, and DNA will stay DNA, but then it grabs its hydrogen. Now, I take this and I put it back in the water. What happens is this. This H is dissociated, and it leaves as a proton. So the proton leaves, and it leaves the electron behind. What does that? What am I doing is I'm putting DNA in water. Notice it's going to happen here. It's going to happen every place where there's a bond. Every place where you've got a bridge. It'll happen here, and here, and here, and here. Each one of those places releases a hydrogen ion. What is it? What do we call something that increases the concentration of hydrogen ions in solution? Acid. Exactly. So, this DNA stands for what? Acid. Acid. This is why. This is why it's an acid. Acid because the hydrogen coming off the phosphate group is releasing the aqueous group. Okay? So it's a strong acid, and it's pretty strong. If you put a big molecule of DNA in water, it's going to release a lot of hydrogen. So it's a fairly strong organic acid. All right, so here's my polymer. That's it. Now the problem is that this is only a two-dimensional drawing of a three-dimensional object. And there is another aspect of this that happens in nature because the bond angles have a particular relationship to each other. When you draw these, remember how you had the bond angles on the molecules last week in lab? And the molecules were bonded at a particular angle. Well, they were true. The bond angles here you can't show in 2 dimensions because they're 3-dimensional. But what you can, if you were to see this in 3 dimensions, what you'd notice is the following. Imagine this. My fist is the deoxyribose sugar. Okay, so my fist is the green sugar. My thumb sticking up here is the phosphate group. My finger sticking up this way is the base. Okay, so here's your nucleotide. And when I make the polynucleotide, what I'm going to do is I'm going to take another nucleotide, again, sugar, phosphate, base, and attach it through that 5 to 3 phosphodiesterase link. Okay, or the phosphate grid. Okay, everybody follow me? Now, the way this is set on here makes it look like the bases are coming out in the same plane. This base is going down, and this base is also going down. Both of them are going in the same direction. That's not true. What's actually happening is this. They're twisted relative to each other like that. They're not quite the same plane. So then when I add another nucleotide, it's also at an angle. And another nucleotide is also at an angle. And another nucleotide is also at an angle. And so you see in the polymer, what you get is like a spiral staircase. The bases look like the stairs on a spiral staircase. So they're all just rotating to each other. And they're rotated always by exactly the same distance. So this angle right here is always exactly the same, each nucleotide out. Yeah. Well, so why does you ask me why does it bond this way and not this way?
Why are they all bent the same way?
Well, they're all bent the same way because they all have. That's a good question. The question he's asking is this: why would they all have the same angle if they're all made out of different molecules? But notice where the bridges form. Bridges are always identical. Yeah. This sugar is the same as that sugar, same as that sugar, same as that. This phosphate, same as that phosphate, same as that phosphate. All the sugars and phosphates are the same. The only thing that varies is this, but that's what's sticking out. So what's really causing the twisting is what's going on here on the sugar and the phosphates, and those are identical for each one. Good observation. Question here first.
On the guanine and the adenine, why is it not exactly why does it have two little chain offs?
Good. You're noticing guanine has a chain off here, and adenine doesn't. And the reason for that is because adenine is missing an oxygen guanine has, and it sets up a possibility for a hydrostatic interaction, which we're going to get to in just a second. Good. Good observation there. That was it. Okay, good. Keep looking for those patterns. These are great. All three of those observations are great. Keep looking. Those are the kind of patterns I want you to try to see in these pictures. Okay, good. Now, so here we are in our head. You see in three dimensions a spiral staircase. That's called a helix. A spiral staircase, another way of saying a spiral is a helix. So these polymers are automatically helices. They're helices. That's common. Whenever you build a polymer, most of the big organic molecules in your body are polymers. Proteins are polymers. These are polymers. Helices are very, very common. They happen all the time because they have to do with the bonding energy. We're going to get into that later when we get into metabolism. We'll talk about thermodynamics and how the bond energies are determining the exact properties of these molecules. Right now, just trust me, this is happening because it's the lowest energy state. But because these particular bond angles are allowing this helix to have a particular bend, it allows this molecule of DNA to do something different than RNA. RNA and DNA are not bent at exactly the same angle. The stair steps aren't spiraling at the same rate. There's a slight difference, and that slight difference has to do with that oxygen that's on carbon number 2 and ribose and not on the oxygen. Again, you can't see why it's using that. But this spiral, the helix that you make in DNA, is slightly different than the helix you make in RNA. So, one of the things that, again, it's hard to see why, but I'm going to try to make you see it. One of the ways that this helix, allows it to form bonds with another DNA molecule in this way. So here, I'm going to make another DNA polymer. And if the polymer has the right structure and is in the right orientation, its helix can helix around the other helix and form what's called a double helix. So it's hard to see in your head. You've got a spiral staircase in your mind. You can see that pretty clearly, I imagine. You have a second spiral staircase, and somehow you take those spiral staircases and make them wind around each other. And that's what the DNA molecule looks like. So in reality, what it really looks like is sort of like this. Here is the first helix, the first spiral staircase. And I'm going to wrap another one around it. So that's a better picture of what we call a ribbon diagram. It's still not perfect, but it's a better sort of two-dimensional image that represents the three-dimensional structure of the molecule. This is more what DNA actually looks like. You have these two helices that are two spiral staircases that are wrapped around each other. And that's why we call it double helix. And the other polymer has to be the right kind. Because you see, these bonds here are not covalent, they're hydrogen bonds. Each one of these bonds where the dots are, not where the lines are, where the dots are, those are all hydrogen bonds. Now, what is it that this polymer here has to be? What's its structure have to be in order to make these bonds? Well, look at this and see if you can see the pattern. Notice how they match. Right, exactly. Notice C always matches with G, and A always matches with T. See that? So that's the structure you've got to have. We call that complementarity. So what that means is this DNA polymer will not just wrap around any other DNA polymer. It will only wrap around its complement. And its complement is the DNA polymer that has a structure such that for every G, it has a C. For every C, it has a G. For every T, it has an A. For every A, it has a T. If it has that structure, then you can still do it. But there's also an orientation issue too. See that? Opposite. Notice how this one is all upside down relative to this one. If you see that, you'll be able to answer this question correctly. My finger is on what atom? Oxygen. My finger is on what atom? Oxygen. See how it's upside down? This is what you refer to as anti-parallelism. So there's two concepts, complementarity and anti-parallelism. Okay, so the DNA polymers must be complementary to each other, and they're anti-parallel. Meaning they're parallel, but one's going the opposite way compared to the other. So they're anti, that's the anti part. Okay, so this is then your basic structure of DNA. You notice any other pattern. One of the patterns. Yeah. Exactly. That's complementarity. C always connects to G, and T always connects to A. The T on the top has an unconnected hydrogen bond. Close. What about this one? Also has an unconnected hydrogen bond. In fact, every AT pair has only two of these hydrogen bonds. How many for CG? 3. Exactly. So there's the other pattern. C and G are always connected by 3 hydrogen bonds. A and T only ever connected by 2. Go down to the hydrogen vent. It's out of the ocean. It really needs to be placed in the talk about later before. These are basically volcanoes that are erupting in the bottom of the ocean. Near those volcanic fields, you have these things called hydrothermal vents, where hot water being heated up by the magma underneath the crust is shooting really, really hot water up. And really interesting. Water coming out of this hydrothermal vent coming out like 2,000 degrees Celsius. And the space is from here to 2,000 degrees Celsius, here it's -4 degrees Celsius. It's not frozen because it's under so much pressure. This isn't boiling because it's under so much pressure. And that temperature gradient exists right here. And in really, really hot water, water that would blow your cells apart, is living a type of single-cell organism that is related to bacteria called archaeans. And those archaeans have a DNA that's different than yours. Their CG to AT ratios are quite different. They live at very hot temperatures. Would you expect them to have more CG pairs or less CG pairs? The third length of DNA than you have. They have more, much more. Exactly. That's one of the ways their DNA can handle that at that temperature you can. Okay, so that's the difference between 3 and 2 makes a difference. Makes a huge difference. Okay, so that's the basic structure. So here is the whole thing all together. That's with all the atoms. Okay? So you got the atoms all listed here, and I want you to memorize especially this structure through here. Okay, so memorize the sugars and memorize the phosphate. Couple of points, not quite done yet. There's 2 other points I want to make. One, like I said, these guys are coming out not in the same plane, which is what gives you that twist. But I said that the RNA twists slightly differently than DNA. And because of that, RNA rarely or is much less likely to go into this double helix form. So you often find RNA that is not double helix. You'll see it oftentimes just as a single strand, a single polymer, as a single helix. It still forms a helix, but it won't wrap around another helix. It will do that, but it doesn't typically do it to another RNA molecule. It will do it to another DNA molecule, or it will do it to itself. But it rarely will form a double helix with a separate RNA molecule. It will form a double helix with DNA, and it will form a double helix with itself. But it rarely forms a double helix. You know, I see a lot of people saying don't over there. So a lot of people staring at me. I'm going to write that down. Okay? So that's the structure. The other thing is notice. Look at these molecules here and tell me, is the 5' carbon above or below the 3' carbon on each of these rival shapes? There you go. Above. So this strand, we say, is running from bottom to top from 3' to 5'. The 3' are down, and the 5' are up. 3' below 5'. The other way of saying that is that end is the 5' end of this strand, this particular DNA polymer, and the 3' end is down here. Anti-parallelism. Notice the 5' are above or below the 3' here.
Below.
5' on top or on bottom? Bottom. There's the 5', there's 3', 5', 3'. So here is the 5', and the 3' at the top. So that allows us then to do this. If I write this, you can tell me what the complementary strand is. I say this is 5', C, A, B, C, A, B, B. Alright? What goes here? G, G, good. G, C, G, K, T, C, G, C, A, A. What goes right here? 3'. What goes here? Again, 3'. What goes here? 5'. The prime is just saying that's the carbon. Prime just means carbon. Exactly. Exactly. And what I said before, I'll say again. Cells read always their DNA from 5' to 3'. Never read from 3 o'clock. Okay, so 5 to 3 will be the next time.
I'm gonna start putting them on YouTube. I'm gonna put all of them on YouTube. Over it now.
That's cool because that was a hard one. I read the Yeah.
Oh, you have it? A video. Oh, nice. That was confusing, but